So this is a snapshot of, we looked at how you can see some different mechanisms in play. And first, they seem like gram negatives do it like figure A here, where they are excreting the quorum sensing signal after it reaches its threshold level of concentration. It'll start diffusing back in and bind with the protein that is then going to go and interact with the DNA and alter gene expression. And then a little bit more complicated one, figure B, is how gram-positive do it, where they have that two component of a sensor kinase and a response regulator. And so the sensor kinase will get the quorum sensing signal docked into its receptor site. And then that phosphorylates it, and then the phosphate group gets handed off to the response regulator, which wakes up the regulator and goes, oh, okay, I need to do my job, which is to go and alter gene expression. So we were looking at this before break, and we talked about how it's gotten more complicated since their initial studies. They now know that there's lots of different quorum sensing signals, and that one organism, one bacteria can have multiple ways of doing quorum sensing. And then where you see these two circles overlapping are the so-called, they call this like the AI2 chemicals are, I'm blanking. It's like translator, any species can interact with the AI2s and understand it, even if they're vastly unrelated species. And so the AI2s were discovered fairly recently. And so we know that there's all these chemicals. We know that there's different reasons for why they're doing this. It could be for bioluminescence, that's where they first discovered it, but it could be for producing toxins to attack. They're pathogenic, could be to produce antibiotics. So we looked at, the idea of bioluminescence. That's where it was first discovered in Vibrio fischeri. And then shortly after that, they recognized that the process of conjugation and transformation, which we've talked about with gene transfer ideas, that many species will use quorum sensing for that. And so the list has greatly expanded in terms of what this can do for the bacteria. Here is the idea that Dr. Bassler talks about in her TED Talk, which if you haven't watched that yet, please do so before the next exam. And so she has a TED Talk on quorum sensing. Oh, and then there's another one I wanted to post that would also be useful. So I'll get that posted. But anyway, she talks about this idea in her video, where if you are a squid or a fish, and you're a prey animal to certain predators. When they're hunting at night, when the predator is hunting the squid, or let's say, they're underneath it. And if you have a moonlit night and the predator is underneath this organism, the squid, they can see them pretty readily if they're not bioluminescent against the moonlit sky as they look up into the water column toward the surface. But if you're bioluminescing, then you camouflage yourself. So if you can imagine a moonlit night striking the water, and then you're bioluminescing, so as the predator is looking up, they don't really see you. And so this is one idea of why they think this symbiotic relationship was established, because it can help the host organism survive predation. So lots of interesting ideas out there, but basically when it comes down to it, what's really happening with all this quorum sensing, all these bacteria doing this, it's a way for them to figure out how many of me are here, like how many, and do we have 10 cells, 1,000 cells, 1,000,000 cells? And really what it allows them to do is, once they figure out through quorum sensing, oh, there's a whole group of us, a big group of us, now we can do a group behavior that will be successful. that we can't do by ourselves, because if you have too few of the cells, it just wouldn't work. And so this group behavior, this beneficial group behavior is kind of a universal. Anytime you look at why are they doing that, it's usually this: they're sensing their population levels. And then that will, once they figure out their population levels big enough, then they're like, okay, now we can do the group behavior. Before break, I was saying, if you think about warfare, if you are a squadron of 10 soldiers, you're not going to attack. You're going to wait until you have 1,000 soldiers there, then go ahead and attack. And hopefully you'll be, have the benefit. So basically, it's this idea of bacterial cooperation, and they actually do use that term, that they're cooperating in order to accomplish a group behavior that they just could not really do successfully on their own. And so this, if you think about it, we talked about pathogens in you, bacteria. If you've had strep throat or something like that, that's what was going on inside you as you experienced that, from the pathogen's perspective. They were successful. They were able to colonize your throat. They were able to access your nutrients by waiting to get enough cells to, there, and then they attacked and were able to degrade your cells and get precious nutrients. So this is kind of that idea where you have, if you had Streptococcus pyogenes in your throat, they're trying to multiply quietly and keep under the radar of your immune system. And then, so they're doing all of this through quorum sensing, and then they're starting to realize, okay, so this is the green light for growth, the yellow light is, okay, we're getting there, and we're sensing there's enough of us here, so now we can go ahead and attack. And so that's when they switch on their virulence factors and are leading a successful attack. So group behavior, in this case, a successful attack. Questions on that idea? How many have had strep throat? Not fun. You ever have it bad enough where you just like swallowing water, felt like knives in your throat? Razor blades, that's how I would describe it. It feels like razor blades. So it is not fun. But that was what was going on inside of you when you were experiencing that. So this is just a cartoon trying to get you to think about the idea that they're communicating before they attack. And if you can imagine bacteria all doing this in the biofilm environment, this could be really nice. And you're all cozied into this biofilm. You're in close quarters. So when you excrete out those quorum sensing signals, it's going to be really nice to be able to communicate that way in the biofilm. They even know that most bacteria have to do quorum sensing just to get into the biofilm lifestyle. But so they're studying this a lot, looking at, how does the biofilm begin? How much of the quorum sensing is involved in that, and finding out a lot. And so they're very curious on, can we disrupt quorum sensing? And hopefully that will disrupt biofilm formation, because biofilm formation can be not so good in many, many scenarios. Yes.
Things that they're secreting would have to be easily diffusible throughout the biofilm.
Yeah, and they're looking at that, like how diffusible are these different quorum sensing signals? They must be fairly diffusible, because it's a very successful strategy for them to do their group behavior in the biofilm. So here again, a big long list of different bacteria and different types of homoserine lactones or AHLs. So these are all gram negatives here, gram-negative bacteria, and then the different phenotypes, like what is it doing for them, whether it be biofilm development or antibiotic production, virulence factor production. So there's a whole range. So I would like you to be able to, come up with ideas on the next exam. If I ask you, like, why would they bother doing this? Think about some phenotypes, like why would they bother to do this? And what advantage could that give them in different scenarios? So all of these different homoserine lactones that you're seeing here, they're, fairly similar chemically. in the sense that they have the lactone ring, and then they have that nitrogen, and then the side chain. And the side chain is really the only thing that's varying in length. And so it makes you think that maybe there could be unrelated species that can hear each other, so to speak, can understand each other's quorum sensing signals, even though they're not identical, but they're close enough. And so that question came up pretty early in this research. And so the fancy phrase they often use is crosstalk. They're cross-talking. And so they were looking at this idea, like, do different unrelated organisms have the necessary proteins that can bind with slightly different homoserine lactones, for example, and can that work for quorum sensing? And they're even getting into the idea of, is this allowing them to tell the difference between kin, where they're, it's all E. coli cells, or non-kin, like E. coli next to Pseudomonas? And is it helping these different bacteria that are in groups to be able to kind of favor their own species, like kin selection? So they're even using these types of terms. So I think I mentioned a while ago that there's this whole discipline of microbiology called sociomicrobiology, where they're looking at these social interactions between microbes and quorum, being able to communicate is a big part of that, of course, the quorum sensing. So here, this cartoon I put up here during the interspecies idea, because all the different bacteria, they're not all the same species. And so just this idea of, okay, can they use, we know they can use quorum sensing to identify how many cells of themselves are there, but what about, can they sense who else is there besides me? So interspecies between different species. And so they're very interested in that idea because they know that most microbes, when they live a normal life outside of the lab, they're living in groups of diverse species. And of course, your gut is full of trillions of cells of diverse species. So, understanding how they're communicating and shifting their behavior. So this was one of the first ones published where they looked at interspecies communication. So this was like a landmark paper where they were looking at cystic fibrosis patients. And they were looking at the idea of interspecies communication, allowing for them to be more pathogenic in the host patient. So Pseudomonas aeruginosa and Burkholderia cepacia are closely related, but they're different enough that they're in their own, two different genera. But they can both colonize cystic fibrosis patients in the lungs and be found right next to each other in the diseased tissue. And they form biofilms, really significant biofilms that can really interfere with the patient's ability to breathe. So what they were finding when they did some careful studies is that Pseudomonas was releasing a homoserine lactone, so a quorum sensing chemical, that actually stimulated Burkholderia to upregulate, in this case, some virulence factors, so they could be even more virulent than if Pseudomonas was not there in the lung. So they noticed that lipases were increased, proteases were increased, siderophores were increased if Pseudomonas was there and releasing its homoserine lactone. So what that was implying is that Burkholderia was able to respond to that quorum sensing signal, and its phenotype shift was to start creating virulence factors. So the lipases break down cells, and proteases break down protein. Siderophores, pathogens like to have siderophores on hand because it helps them latch on to iron. It's a way for them to chelate iron in their host so that they can scarf up some iron. A lot of pathogens have a high iron need. So this, the only two I want you to look at here are where I've drawn arrows. But here you're seeing upregulation of siderophores here. So this is on the y-axis in siderophore levels. And this is if Burkholderia was exposed to the homoserine lactone made by Pseudomonas. You can see a huge increase in siderophore production. If you compare that to the control, where it was just Burkholderia with no access to the homoserine lactone from Pseudomonas, no siderophores. So this was one of the first published studies to demonstrate this interspecies, and importantly, in a very serious ecology here, looking at cystic fibrosis and, you know, how to better understand it so that you can have the patients live as long as they can, as healthy as they can with this terrible disease. So they've even found that you can get, so the previous example I just talked about were 2 gram negatives. So we also know Staph aureus can be in the lungs of cystic fibrosis patients. Staph aureus is gram positive, Pseudomonas is gram negative. So then they were able to show that you can have very different cell types, and they're still able to understand the quorum sensing signals between them, and that can help them to be more effective as pathogens in the lung tissue. So they're really keying in on this idea, and they're trying to think of ways to successfully block this communication. Just like in warfare, if you can block communication between the units, where they don't know where each other, you know, where you're located, you can't coordinate. If you can block that communication, that's an effective way to try to reduce their ability to fight effectively. And so again, with this, with the pathogens, if you can block their ability to talk to each other, hopefully the disease will not progress. They've looked at this a lot in plants as well, and they're finding that bacteria that hang out on leaf surfaces of plants, you can see that they actually are producing homoserine lactones that actually cause pathogens to not be as effective. So here you have this idea of the good bacteria on the plant is able to kind of ward off the bad bacteria. And they think it's by basically a mimic compound, the AHL or homoserine lactone is kind of like a mimic compound. But the big picture idea is that these beneficial bacteria are messing a little bit with the pathogen's ability to communicate. And so then the pathogen, Pseudomonas syringae, cannot attack. So this interspecies communication was shown to be a thing, and then Dr. Bassler's team discovered the AI2 family of chemicals, where they call that there it is, universal language. I was blanking on that phrase. So when she first published this paper back in 2005, she was finding AI2 signals in half of the bacteria that she had on hand in her freezer. And so there's been a lot more work since then, and she's finding that the AI2 signals are very common, to the point where she refers to them as the universal microbial language. It's a language that, you know, if you're speaking French or Italian, you can still understand the AI2. And so that's the basic idea here. So turns out the more they look, the more they realize there's a very sophisticated way that microbes can communicate with each other, even unrelated organisms. And then they can, each one could have multiple ways of doing this. And so, I mean, I'll just give you a few examples of some ideas here where the either to manipulate or cut the quorum sensing process. For example, there's a red algae that lives in the ocean that if you're an algae living in the ocean, and red algae tend to live a little deeper in the water column where the sunlight isn't so strong, but they're photosynthetic. They want to do photosynthesis. That's how they get their sugars and their ATP. So if they're covered by a thick layer of biofilm of bacteria, that's not very good, because then they can't get good access to sunlight. They're already in a dim environment. And then if they get coated with this thick goo and then a bunch of bacteria blocking or shading their leaves, they're going to have a harder time doing photosynthesis. So what they found is that the red algae is actually producing a substance, an autoinducer that actually cuts down on biofilm production by these bacteria that hang out on their surface, primarily some gram-negative ones. And basically what they see is that the red algae are producing this mimic. It's a uranone type of chemical It's very similar chemically to homoserine lactones. And it's binds competitively so that it takes the place of the homoserine lactone in the receptor of the protein receptor. And so basically it cuts the ability of the gram-negative bacteria to effectively do quorum sensing. And so Serratia is the gram-negative bacteria that the red algae is trying to keep at bay. And then what they find is that it allows some other bacteria to come in, but they're not as abundant and they don't cause as much shading. So the red algae wins in the sense that the red algae now has a better access to sunlight and is not so heavily colonized. So that was one of the first examples that they gave of how, here you have a plant, which is very different than Serratia, and yet they're able to mess, or at least the plant is messing with Serratia's quorum sensing. And so they are finding distantly related organisms can do that. Here in your gut, this could be going on if you ever get exposed to Vibrio. So Vibrio cholerae, of course, causes cholera. In your gut area, what they're finding is that certain bacteria that are just hanging out in some people's guts will produce a quorum sensing like chemical called DPO. And that actually limits Vibrio's ability to colonize the gastrointestinal tract. So you definitely would want something like that going on if you get attacked by Vibrio. That can be a very serious, debilitating, and deadly disease. And luckily, we don't have too much to worry about in this country, but in many areas of the world where that is a serious problem year round. Okay, so going back to plants, they can mess with bacteria that are hanging out with them. They can recognize quorum sensing signals from bacteria. They can produce mimics, quorum sensing signal mimics. They can break them down. And they can actually stimulate quorum sensing. So this, again, very different organisms, different domains of life, and yet they're able to communicate or stop communication between the plant and the microbes that are in the area. With this idea of stimulating quorum sensing, it can be a positive or negative thing. I'll give some examples of some of these ideas. A lot of the research that's been done are on agricultural crops because we care about what have enough food. So they've been looking at things like pea plants, rice, soybeans, tomato. And when they look, they find that there's this quorum sensing process going on in this interaction between the plant and nearby microbes. And it could be beneficial microbes or pathogens. So here, this one's tricky. So if you imagine that you're a plant and you have a pathogen present, maybe it's in the soil by your roots. And of course, if you think about warfare, the plant will get in trouble if that pathogen's numbers increase, where it can be more effectively attacking the plant. So what happens is the plant will actually stimulate quorum sensing in the pathogen. So basically, they try to get the pathogen to show its hand before the pathogen has amassed a huge population. So if you can trick the pathogen into showing up, so to speak, where the plant can detect it, then it will be able to, the plant will be able to kick in its defense earlier before the pathogen had a chance to amass an army to attack. So it's a really sneaky way for the plant to kind of assess, are there pathogens around me? And can I get them to show their hand so I can trick them into this ability to, essentially the plant is outsmarting the pathogens through the quorum sensing process. And so they've demonstrated that the bacteria can, the plants can do that against many different bacterial pathogens. And so it's a handy little strategy. As far as if you have beneficial bacteria hanging out with you, you may also want to get their quorum sensing up and running. And whoops, that can be a good thing because it benefits you ultimately. So the plants often will do that too, where they will promote quorum sensing in beneficial bacteria that are coexisting with them long term. So there's been lots and lots of published papers looking at this idea. Here, one published on plant beneficial gram-negative bacteria, interkingdom signaling between plants and pathogenic bacteria. So lots of different studies going on, same, looking at animals too, of course, with humans and other animals looking at quorum sensing of pathogens in particular. So here they're looking at the idea of bacteria hanging out in your guts again, and that the human is also interacting in this quorum sensing process. And so the idea that you can have quorum sensing going on, they have established this pretty well with pea plants that hang out with rhizobium bacteria. The rhizobium are busy doing nitrogen fixation for the plant. And so the plant really likes that. I mean, that's a huge benefit. And so they encourage this association. They attract the bacteria to their roots, and they use quorum sensing to get into this relationship. And so that has been established for many years now. And now they're taking a lot of focused look now in the human gut and looking at what could be going on. Could the human host cells be excreting out any kind of quorum sensing signal that some of the bacteria in the gut respond to? So here they're showing this idea of an AI2 mimic being released by the human epithelial cells. And it is influencing the bacteria and their ability to do quorum sensing. So they're looking really carefully at this, especially at some of the that more and more they're looking at some of the quorum sensing going on in human pathogens that aren't really causing overt disease, but they're causing some subtle shifts in the host that are long-term are not good to have going on. So here they're looking at antibiotics. So if they did a study where they had, I think this was a mouse study, where they had the mouse take antibiotics, and you can see a shift. So after the antibiotic treatment, the green and blue, they're trying to show us a nice balance here of the about equal numbers. And then after antibiotic production, it wiped out all the green ones. And the green ones are the AI producers, which is that universal quorum sensing signal. And so finding that this was not a very good balance for the host. So then what they did was they gave a probiotic that had E. coli that could express that AI2 since it was missing so much. And when they did that, they were able to get the balance back in terms of the original balance pretty much of before the antibiotic treatment. So this is something that they're very interested in, because when you take antibiotics, this is happening. You are wiping out, especially if it's broad spectrum antibiotics, you're wiping out huge numbers of microbes in you. You have to be really thoughtful about taking antibiotics. And you should always ask your doctor, is this a broad spectrum antibiotic or narrow spectrum? And if they say, well, it's broad, then you should say, is there a narrow spectrum available? Because narrow spectrum, the analogy they often use, narrow spectrum is like a rifle bullet. Broad spectrum is like a shotgun, where you destroy so many of your beneficial microbes with a broad spectrum. And I would say probably 80% of the antibiotics that are handed out are broad spectrum. And so you are significantly altered in terms of your microbiome. We'll talk more about this when we get into the human microbe interactions, but it's something to be careful about. I'm not saying never take antibiotics, because sometimes you have to save your life, but you have to be careful. They're even looking at viruses and that attack bacteria. So bacteriophages, this is Bonnie Bassler again, her work, where they're looking at, now they're finding phages can do this too. Phages can do quorum sensing, and bacteria can do quorum sensing to detect phages. And so they're seeing it both ways, and they see that bacteria are using quorum sensing to defend themselves against the phage attack. So they're, the more they look, the more they find. So in many cases, from a human perspective, we want to quench the signal. If you have pathogens attacking you, you want to stop them from communicating, because if they can't communicate and figure out, are we at the population level where we can attack? If they never get that information, they won't attack. That'd be great, because then you wouldn't need antibiotics. Your immune system will catch up quickly and defeat it hopefully. So this idea of quorum quenching is a hot topic right now. They're looking at this idea because, as you know, unless you've been living in a cave, antibiotic resistance is a big thing. And there are plenty of concerned microbiologists out there that are saying we need something different. And so this idea of quorum quenching to reduce pathogen disease is a hot topic. So basically you're trying to disrupt. So they're even looking at this in plants. Cannabis is one of them, where they're looking at the normal microbes that live inside the plant. So those are endophytes. And the endophytes are actually using the strategy of quorum quenching in terms of trying to protect the plant against pathogen attack. And so this idea of Chromobacterium violaceum is a pathogen, a plant pathogen. And so they were able to find that the endophytes were messing with the quorum sensing of Chromobacterium. So they're finding more and more of this ability in the human mouse model plants, that the good bacteria hanging out with the plants are able to do this. So here they're looking at the endophyte idea. Could we mine endophytes? Could we look for endophytes and figure out what their quorum quenching chemicals are? So this is a hot topic right now. This is just the flow chart showing the basic idea of how they would go at that. I'm not asking you to learn that, but if you're curious, this is how they approach this idea, where they can finally purify the quorum sensing chemicals or quorum quenching chemicals. So it's a hot topic right now. Here they're looking at quorum sensing disruptors again with the endophytes. So if you're not aware, a lot of the pesticides that have been used for commercial egg production have been phased out. They've been outlawed or banned. And so they're trying to figure out other ways to protect plants against pathogens. And so this idea of, can we use the good bacteria inside the plant to quench the quorum sensing of the pathogen? So they're finding it in humans, the epithelial cells, which are in your mouth, your gastrointestinal tract. I think the study they did for this one was the mouth. They were looking at Pseudomonas aeruginosa, and they could show that they release a quorum quenching signal. So that made it much harder for Pseudomonas to get any established population. They're finding soil bacteria can break down the quorum sensing chemical. So again, if you can destroy the chemical, that would be good. You're essentially quenching their conversation. So they're finding that they can attack it and break it down. Bacillus and Streptomyces can do that. So there are different strategies to quench the signal. And so they're looking at all these different options, because if you can stop the communication, the pathogen should not be able to gain any kind of traction and cause any problems. So maybe the bacteria that are breaking down the homoserine lactones, they're just using it for nutrition. I mean, that would make sense. It's a nutritious looking chemical. So it could be just something like that going on, but the hope is we can find more, pinpointing, scrambling of the communication so that they can, develop these chemicals to quench. So they're definitely looking at ways to interfere with quorum sensing when it comes to pathogens, whether it be a human pathogen or a plant pathogen. And so with the issues, all the issues we have with antibiotics not working anymore, this has become a really hot topic. So this is a review paper. Again, just trying to show you that they are discovering a lot of different chemicals that are potential for upscaling and scaling up and doing some more focused human studies in particular. So lots of ideas here. This is just showing some different strategies maybe of how they could quench the signal, either modifying it, degrading it, or somehow allowing to it, get it sequestered so that it can't actually physically work the way it's supposed to. So a lot of this is still going on in mouse studies and rat studies, but they are making progress and they have done a few human studies, but it's going to take a little while. I think in your lifetime, you'll see it as a drug treatment. If you go to the doctor and you're sick with a bacterial pathogen, I think in your lifetime, you'll see that there will be quorum quenching opportunities instead of antibiotics. And then there was this question that I got asked, I didn't know, so I did have to dig a little bit. Can bacteria quorum sensing signals impact our immune system? So could the bacteria that are in you, pathogens or non-pathogens, could they somehow mess with your immune system in some way? And the short answer is yes. So Pseudomonas, if you get Pseudomonas in you, it's typically a pathogen. It acts pathogenically to you. And here they're saying that, yeah, it is suppressing the immune system response. They've done some of this in lung tissue, but they've even done it in mouse environment. And so it seems like that's a possibility. So they are definitely on the trail trying to figure this out more deeply. Yeah, sometimes your body will produce the right types of cells and memory cells that can remember that pathogen. Sometimes not. We'll talk a little bit more about that in the human microbe interaction, where some microbes you can get sick with over and over and you never build an immunity to it. And it's often the microbes like food poisoning microbes that you ate something bad and, you know, probably several of us in this room have had food poisoning more than once. And it might have been from the same microbe, but. Okay, switching topics, but some of the basic ideas are still in play here of communication. So after quorum sensing came along and they discovered that, then not too far in the past here, they came up with this discovery, nanotubes. Now, this is an interesting comparison to quorum sensing because if you think about quorum sensing, the microbes that do quorum sensing, they're sending out their quorum sensing signals out into the environment. Like anyone who's there could hear it, so to speak. With nanotubes, it's a system in place where it's a very private conversation between one cell to the next. And so it happens because of this creation of a nanotube, which is just the cell membrane of one bacteria starts to extend out into this long skinny tunnel and hook into a nearby bacteria. And then through that tunnel, they can pass information or molecules. It's not the same thing as a pilus. we talked about a sex pilus and how that can be used. It's not that. It's actually cell membrane material that will, whereas a sex pilus is proteins, subunit proteins that are built into a tunnel. So this was one of the first landmark papers back in 2011 to publish on this. And so basically, it's a very private conversation where you have, these are actual cells under the microscope where you can see the nanotubes connecting some of the neighboring cells. And here with this mock-up, they're showing how you can have different things past some stretches of DNA or plasmid, certainly, different proteins or enzymes. And so when they discovered this, I think the first thing that they noticed was a visual, like, hey, what is that? They discovered that first and they thought that was odd. And then they started looking at it more carefully and realized it wasn't a sex pilus or a pilus in general. It was actually cell membrane material. And so, but basically it's like a tunnel. And it's just cell membrane material essentially. And they've done a lot of, lab studies like, okay, so why are they doing this? Why would this be a thing? They're bigger than pili, so that's interesting. So that means more things can pass between cells this way because it's just a wider diameter. And they are finding it in the domain bacteria and domain archaea, both domains. There's species in both. So intercellular molecular trade is a good way to think about it. Here's a mock-up from that paper where they're showing a variety of different things. They're saying, a bunch of different molecules can get transferred, plasmids can get transferred. And so basically, when they've done lab studies, they show these cells will start making these nanotubes and they'll just kind of get it out there and it kind of randomly, kind of floats along until it hits another cell and then they form a tunnel between the two. And so it is pretty amazing to watch this process. And so they're like, okay, what's going on here? What's getting transferred? It's definitely small chemicals. It's a pretty wide diameter, so you can have a range of different chemicals. So it could be nutrients that are getting transferred from one to the other. And they've done some nice studies with that, where they put the microbes under starvation and watch how they cooperate and like share nutrients with each other. Or there might be proteins passed, messenger RNA they've seen get passed, DNA gets passed, plasmids, viruses, bacteriophages can get from one cell to the next through nanotubes, unfortunately. So that's another way for viruses to spread from bacteria to bacteria. Toxins. So they've done some studies where one bacteria sends a toxin through a nanotube to the other bacteria to kill it so it can extract all of its nutrients. So it sounds like something from a sci-fi movie, but at least in a lab study, they can show that there can be cooperative things going on where they actually help each other, but also attack going on where they are definitely causing problems, one against the other. Okay, so we'll continue on with nanotubes on Wednesday.